Hello, everyone. On behalf of Understanding Ag and the Soil Health Academy, I'd like to welcome you to today's webinar. Don't buy it, grow it. How to make the nutrient cycle work for you, with Understanding Ag partners, David Kleinschmidt and Shane New. We will be taking questions and answers at the end of their presentations, so please be sure and type your questions into the Q&A box. With that, I am not going to spend time introducing Shane and David. We'll call on Understanding Ag partner Shane New to kick it off. Shane?
Thank you, Gabe. Like Gabe said, we'll be addressing questions after both of our presentations. Kind of bear with us in case some technical difficulties. We've got a lot of wind here this evening. And at the end of my presentation, we'll be sharing David's screen, so we'll have to stop and convert over. So welcome everyone for tonight's webinar, How to Make Nutrient Cycle Work for You. You know, this is probably one of the more difficult.
Excuse me, Shane, we can't see your screen.
Hold on. Just give me a second here, please.
Maybe a good thing that it's taken you a few minutes. We're getting a lot of people joining us here, which is really good to see. We really appreciate all of you taking the time to join us on this webinar.
Obviously, been through this a dozen times and obviously some technical difficulties, but give me a second here. Are you able to see my screen now?
There, it just came up. You'll just have to start the slideshow and you can take off.
Are you able to see that now, Gabe?
Perfect. Thank you, Shane.
Apologize, everyone. So how to make the nutrient cycle work for you? You know, it's more important now to start building resiliency into our farms and ranches. You know, it's probably one of the more difficult presentations to put together. You know, I want to try to address this looking at the principles. And we always talk a lot about context when we talk about the 6 principles. And obviously over the last 3 years, you know, we've seen a lot of supply chain issues, energy supply issues, and also, you know, to acknowledge the war in Ukraine. And there's been a lot of disruptions occur and how it's affected us, you know, and a lot of our input costs in agriculture on both our farms and ranches. So, you know, when I was getting this presentation put together, you know, we always talk about energy cost and how it reflects anhydrous ammonia cost, because nitrogen production consumes about 1 to 2% of the world's energy. And, you know, we've always put there's kind of a correlation there, but this research done by Texas A&M kind of debunks that. You know, really the price of anhydrous ammonia is more directly related to commodity prices and not so much reflective of natural gas prices. So this is a pretty good little graph I found, and I kind of thought, you know, we as farmers and ranchers, you know, how do we become more independent and quit being so dependent upon some of these inputs and some of these things that's kind of out of our control that's occurring? So why is nitrogen important? Well, it's needed by every living cell. That's why it's important. You know, nitrogen used in many molecules, just some of the overview here, nucleic acids, DNA and RNA, the building blocks of life, protein structure, think of cell walls, our own bodies, muscle, and also chlorophyll within inside the plants. We rely on pools of ammonium and nitrate in the soil that can be taken up by plant roots, but these pools are really relatively small. And these are the kind of the pools we focus a lot on, the ammonium component and also the nitrate component when we talk about nitrogen. But the forms of nitrogen that are in our soil is kind of interesting. About 20% of the nitrogen in our soil is nitrogen gas. Ammonium about 0.2%, nitrate about 0.4. Plant nitrogen, now this is decomposing residue, plant roots, for an example, is about 2.2%. But this organic form of nitrogen in our soil is about 77% of it. Now, I just want to acknowledge here, this is research done by Dr. Stephanie Yarwood at the University of Maryland, and this is over many systems. So it's going to vary a little bit, 1 or 2% here or there, over different types of systems. But this is an average of different systems. One thing I also want to kind of point out is this total nutrient digestion test. This is a 12-inch test looking at the forms of nutrients that are in our soils. And we're talking about nitrogen here specifically. So on this particular sample, we have 6,696 pounds of nitrogen. Now, this is both in the organic, which means it's bound to carbon, and also in the inorganic form that can be utilized by the plant. Now, all this is in the inorganic form, obviously. Most of this is bound up to carbon, so it's unavailable to be utilized by the plant. And these are the forms of pools of nitrogen in the inorganic form, in the organic form within our soils. And this is the one we need to kind of a little bit focus on. I just want to kind of highlight these a little bit. Part of it's, that nitrogen source is from DNA, from living materials, be microbes, plants. A larger fraction of it, obviously, is proteins. Proteins are built up of amino acids, labile carbon, nitrogen in our soils. These are very simple structures, 1 or 2 carbons. There's also a percentage that's unknown that we really don't know. We know it's bound to carbon. We really don't know how it's utilized within the soil. There's these very complex structures of carbon and nitrogen in the soil, these aromatic rings. We also have these amino sugars such as chitin. Remember fungi produce chitin. Not only are they helping utilize enzymes to dissolve this organic portion of nitrogen, but also their cells themselves are a form of nitrogen that gets remineralized within the soil. Organic nitrogen must be decomposed to release the ammonium that can be taken up by the plants. So obviously we've got to have this organic matter, this organic form of nitrogen that can be broken down. But what's breaking it down? In order to make it in the form of ammonium and nitrate that can be utilized by our plants. It's the microbes. Compounds like proteins are broken down by soil microbes utilizing enzymes that break down these protein structures, converting it back into ammonium, then also into nitrate that can be utilized by the plant, and also the ammonium can be utilized back by the microbes. And I just want to show you a quick illustration. I don't want to spend too much time on this, but basically these protein structures are pretty complex, but it's the microbiology, the bacteria and fungi that are producing the enzymes, breaking these structures down into simpler structures. So think of this, as a building, and the enzymes, protease is being utilized to break the protein down into peptides. So basically think of this as the walls of the structure. Then they're utilizing peptidase enzyme to break it down even further into amino acids. It's still in a carbon form here, but then the biology's breaking it down and putting it in an inorganic form that can be utilized by the plant and also by the soil microbes. This is all done by enzymes produced by the microbes. So basically the nitrogen cycle, it's taking the plants, putting nitrogen in the organic form, in organic material, being decomposed by the soil microbiology, converting it back into ammonium and then back into nitrate. And notice how this ammonium, you see the arrow going back towards microbes. These nutrients, especially ammonium and also nitrate, can be utilized by the microbes. Then basically the microbes, as they die, return back into organic form of nitrogen. They have the nutrients immobilized in them, and it can be cycled back. So it's a cycle. And that's what we're needing to try to get functioning back within our soils. So we need to start thinking about the principles, how these principles matter. How do we start getting these principles on our farms and ranches in order to get these nutrient cycles to occur? So why is tillage so destructive? When we talk about principles, we talk about minimal to no soil disturbance. Well, tillage does a lot of things. One thing it does is, promotes a lot of nutrient runoff. It also disrupts the soil microbiology. Remember, it's this biology that's helping do the decomposition, break down these complex molecules, and utilizing those nutrients, converting it back into plant available forms, such as ammonium and nitrate. Increased residue oxidation. So basically that organic matter and stuff that we're trying to hold in our soil, it's oxidizing it, being released back in the atmosphere and not being captured back within our system. The next principle, when we think about this, living root in the soil. We want a living root in the soil as long as possible. What's the living root doing? Well, the living root is capturing energy, sunlight, and water and carbon dioxide to make sugar, glucose. But it's also taking in ammonium, nitrate, and amino acids to help build cell wall structure, proteins. So why would we ever want to take an opportunity to waste any energy? It's the reason why the living root's so important. And here's a good example. This was taken 2 weeks ago, but look at how these fibrous roots on these rhizosheaths are just full of biological activity occurring. Think about when I talked about just a little bit earlier about it's the biology producing the enzymes, breaking down organic matter, converting that organic matter down into proteins, acting upon it, and converting that protein into inorganic nutrient such as ammonium and nitrate. What happens when we put too much nutrients, synthetics, into our system? This root's not sending out the chemical signals to make the association with the soil microbiology. So obviously this plant's becoming very dependent upon inputs in order to thrive or prosper. And we can notice this by how clean these roots are. There's not a lot of root exudates being produced. So we're not, basically we're writing checks here. Here's another good example, during the growing season last year of a lot of nitrogen that's been applied. The plant's not really having to make the work relationship with the microbiology. Here's another good example where we've got good rhizosheaths covered with soil microbiology. Look at the nutrient cycling. When we start talking about not only the nutrient cycle, but look how this aggregation is starting to help with the water cycle. Building that aggregation, allowing water to infiltrate deeper into the soil, allowing this microbiology which works in a subaquatic environment in order to produce those enzymes to help decompose, to help cycle those nutrients. Cover on the soil surface. Now, why would cover be so important? Well, a lot of this is, obviously is a living plant that's going to start to decompose, cycle back into the soil, but also more than that, it's helping protect that soil, help, as I stated earlier, allow this microbiology to work in this water film subaquatic environment. And also water is extremely important for hydrolysis, breaking down these chemical bonds, allowing the enzymes to work, function, to break down these structures in order so we can cycle the nutrients that our plants need and also the biology needs in order to function. And here's just a good illustration. This is underneath the microscope. All these little circles here, these are bacteria. There's a little fungal hypha right there down here in the bottom center. But all this brown material, I mean, this is decomposing what used to be organic matter, now being broken down by the biology, converting this organic form of a nutrient, breaking it down, converting it into an inorganic form, making it now plant available. You know, the biggest thing, if you take anything away, we've got to start understanding it's the biology that we need in order to make this process work. And I just want to give you a good example. When we talk about a carbon bond amino acid, this is what it structurally looks like, the composition. Notice a nitrogen group here, carboxyl group, but it's bound to carbon. This is what we want. We also need this carbon for the microbes, but also this nitrogen is what we're looking for to convert into the inorganic form. So when we talk about, you know, nutrient cycling, and, you know, a lot of people, when we visit with them on farms and ranches, you know, tell us, you know, they don't have time to plant a cover crop, or they, you know, they don't understand why it's important to keep a living root in the soil. So here's a good illustration from last spring. But just look here, this is a cover crop. So basically we took a 3x3 square, clipped the cover crop, sent it into Regen Ag Labs, and basically get a nutrient analysis of the nutrients that were tied up in that biomass. Look at the nitrogen, 245 pounds of nitrogen per acre that was tied up in that vegetation. You know, the only way we're going to cycle that back is via the biology. So diversity of plants. So when we think about principles, you know, diversity is pretty important. You know, one thing with diversity, we're keeping the living root. We're also creating, I started thinking more about the architecture of the roots and how it's feeding different soil microbiology, you know, producing different carbons, sugars, attracting the soil microbiology, the microbes producing enzymes, and dissolving nutrients from protein structures, also nutrients that are bound to silt, sands, and clays and minerals, and making them from the organic form into the inorganic form. So the reason why diversity is important is because of the different rooting architect structures, but also the different carbon compounds that each one of these plants produces, feeding different soil microbiology. So I know most people have seen this diagram talking about the nitrogen cycle. You know, I just want to acknowledge it a little bit. You know, so obviously we have legume plants, and it is the plant, the legume makes an association with rhizobium bacteria. The rhizobium bacteria are fixating nitrogen for the plant into ammonium. It is taking the biology, the bacteria, the nitrogen fixing bacteria, also through enzymes, turning that ammonia into ammonium, then also going through another process of breaking it down into nitrates, back into nitrites, back to where the plant can reutilize it again. This is a cycle. The plant obviously grows, it decomposes, breaks down. Also other creatures decompose, converting back nitrogen, carbon, back into the cycle again. Animal impact. You know, animal impact, you know, very important. You know, the nice thing about the animal impact, we're utilizing the animals for an economic gain, obviously, to forage some of this biomass, breaking down some of these cell walls through the biology within the animals. And the animals are basically converting that, as we well know, the excess back in the form of manure. And the biology in the soil will help break this down even further, reassimilating it and putting it back into forms that we can be utilized either by the cells, the biology, and or by the plant. So do the principles really matter? Yes, they do. One thing when we work with farms and ranches, we really encourage everybody to do their own testing. We encourage everyone to utilize the Haney soils test. This gives us some really good information of where we're at in the system. One thing we also encourage, we could go on this nitrogen cycle really in depth talking about the predators such as protozoas, nematodes. They're also helping cycle nutrients in our soils. But I wanted to keep it really brief tonight. David's got a great presentation talking about a lot of the economics, but making an acknowledgement is, most of our soils don't lack the nutrients we need to grow crops. What we're really lacking is a soil microbiology to help convert that organic form back into an inorganic form that can be recycled back within our systems. David, are you ready?
You bet. I'll get fired up here. Let me see. All right, can y'all see my screen? You hear me good, Shane?
Yeah, David, I hear you well.
All right, so Shane did a fine job of explaining a lot about the nitrogen cycles here. I'm going to talk a little bit about, how to utilize our cover crops that we might have out there. And also, thinking about how should we manage our nitrogen applications. No matter if nitrogen's expensive or cheap, we should be thinking always about how to manage it so we get the greatest return on investment for that pound of nitrogen applied in return as a bushel of grain. So one of our biggest challenges in agriculture is that still too many people lack this understanding of a biological nutrient cycle, as Shane was explaining there. We still base our nutrient applications and management on a chemical-based nutrient test. Basically, especially here in the United States, we go out and we pull a soil sample and we look at it and say, okay, first off, most guys would just look at pH and say, do we need lime or not? Where do we stand with P and K as far as phosphorus and potassium? And on our test here, we don't even look at nitrogen. So we have no clue of what might be in the soil. We just go ahead and know that, hey, for 200 bushel of corn, we got to apply 220 bushels of nitrogen. That's kind of how we manage it, which is, quite frankly, very sloppy. Another common myth about inorganic fertilizers is that we are feeding the plant directly. Well, that plant's only able to take up only about 10% of that fertilizer directly. The remaining has to go through a microbe's gut, the other 90%. But of that pound of fertilizer, that 100 pound of fertilizer, however much we're applying, at most only about 60% actually gets into the plant. So if you think about it, if we apply 100 pounds and only we're getting 60% in the plant, where's the other 40 pounds going? Well, it's either locked up in the soil or basically it's locked up into a microbe's gut. That's why when Shane's talking about the microbiology, we have to think about, how do we get the microbes to be able to start cycling these nutrients? And so one of the things here is I worked in retail. When I worked in retail, a lot of guys would focus on the Liebig's law of the minimum. And this was basically stating, whatever that stave on that barrel is, whatever's the lowest stave is where most of your yield potential loss is going to occur, whether that's nitrogen, potassium, phosphorus, calcium, zinc, manganese, boron, whatever, water. The one thing this never did take into consideration really was the factor of what does carbon play in that soil? And also, what about your microbiology? So it just looks at the minimum. So if we were to look at a soil test or a plant tissue test, we'd go through it and say, what's my lowest one? And we'd say, okay, well, if you know, zinc's low, then I need to apply zinc. But we also have to understand that there's, oh, I think I froze up here a second. Hold on. Okay.
No, you're good, David.
There we go. It got back up on my side. So there's this law of the maximum. And what this is basically stating is that some nutrients are synergistic to each other and others are antagonistic. So when we're looking at that, either soil test or tissue test, we need to look and say, you know, if one nutrient is limiting, is it because that we have an excess of another nutrient out there that could be tying up that nutrient? So it doesn't matter how much of that zinc that we might apply, if, you know, let's say calcium is our limiting factor or something, or we have too much calcium or too much phosphorus there, you know, hey, we could be tying up zinc. So applying more zinc won't matter until we can get that phosphorus in line. And then there's this Sir Albert Howard's law of return. And this was, if you did do any research about Liebig, his last book was basically, because his first book was on that organic matter really didn't matter. And it was all on the chemical composition. But his last book, before he passed away, was talking about how that actually taking crop residues and having them being put back on the soil and everything was this return, and it increased the organic matter, which increased the productivity of that next crop. Well, what we were figuring on was, you know, this was back, you know, early 1900s, late 1800s, is that it's due to these microbes in the soil. You know, we're building that aggregate, we're protecting the soil, you know, doing all the things that the principles that Shane was talking about. So this picture here happens to be a cover crop from a prevent plant in 2019. This was planted in July, which would be about the same time that a warm season cover crop after wheat harvest could be planted in my area. You know, we were extremely wet that spring, and then it just turned off fairly dry, you know, be about average for our July, August, September timeframe. But the cool thing was, you know, Shane was talking about taking that biomass sample. So I did that as well. Took a 3x3 square. And what we could see here is we had 7.8 tons of dry matter in a short period of time between July to about 1st of September there. We had 279 pounds of carbon, 146 pounds of phosphorus, 654 pounds of potassium. And the standard soil tests on this did not show that we had those levels. So that kind of threw me for a loop and had me start really asking questions. The biggest one was 279 pounds of nitrogen in here. We had not applied any nitrogen onto this field or any fertilizer since the spring of 2018. So I was like, where did this nitrogen come from? That just blew my mind, right? So if you were to look at this today, and this cover crop mix, you know, back then was about $25, and it was, I don't remember, an 8 or 9-way mix. But if we were to look at the economics of this today, and this is at today's fertilizer prices, so to pay for 280 pounds of N roughly is $308. 146 pounds of 046O would cost you about $138. 654 pounds of potash would cost you $438. So that's $884 out of a $25 cover crop nutrient value. Just imagine if we put another cover crop in behind that and we were able to sequester those nutrients and hold it for that next cash crop, what we might be able to achieve and how we can really start reducing the amount of fertilizer we need to apply and, you know, start saving some money. You know, now everybody talks about these carbon markets as well. So if we were to say carbon's worth about $60 a ton, that's another $180 an acre value. You know, so, you know, $1,000 pretty quick right there that we could figure on. So when I was talking about these nutrients and where they came from, this total nutrient digestion, and this is only looking at that 6-inch sample where we're looking at the organic and inorganic pools there. There's 2,300, almost 2,400 pounds of nitrogen, 640 pounds of phosphorus, 2,100 pounds of potassium. And just in the 6-inch profile there. Shane was showing some earlier in the 12-inch profile. So when you look at that, you have to say, my gosh, why do we really need to fertilize? An interesting thing too is, which is every state has, you know, some sort of agronomy handbooks or whatever. Illinois' agronomy handbook, I can't remember the reference number on it. I've got it in another presentation, but the phosphorus test on a standard soil test, if we were to look at the usefulness, reliability, and efficiency of these tests, the phosphorus test is only about 85% reliable, and the potassium test is only about 60% reliable and effective. So a lot of times we're basing our management decisions on how much fertilizer to apply on something that isn't 100% or even close to it. So, you know, we're willing to waste money on something that we really don't understand and don't know. And I've had a lot of farmers tell me that they've applied a copious amount of potassium or potash trying to raise their potassium levels in their soil and never could achieve that. You know, you might see a rise in the base saturation, but they couldn't achieve an increase in their actual potassium levels in their soil, which that goes into a whole problem of salting out the soils and having that salt be a lookalike molecule being taken up by the plant, causing all kinds of deficiencies and a whole host of other problems. So the thing we need to understand about cover crops and breaking down is this whole, our carbon to nitrogen ratios of those cover crops, and understanding that the ideal microbial diet is about this 24-to-1. So anything that is below that would be a quicker decomposition rate, where anything above that is going to be a slower rate. Now, we also have to understand that soil microbial activity in that soil is always going to be different. You take a soil that's got 150 parts per million CO2 respiration, it's really cranking, where if you got something that's in the teens, you know, it's going to need nurtured along. You know, you just can't throw a big carbon cover crop out there because it won't be able to handle that. So we have to understand how this plays and how to mix each one of these together for our cash crop needs based on which one's going to be releasing. So some examples of a low carbon and nitrogen cover crop, you know, or even cash crops here, you know, soybeans, clover, alfalfa. Those are some, you know, think of these as your low carbon to nitrogen ratio plants. So whenever we look at like soybeans, they're low carbon, high nitrogen. So really what we need to do for that soil and for that plant is, in front of the soybean, have a high carbon, low nitrogen cover crop. Well, on the other side, we have, you know, things like wheat, cereal rye, triticale, barley, corn, milo. Those are your high carbon to nitrogen ratio plants. They are high carbon, low nitrogen. So if we were to take a sample of this, you know, that corn might be at a, you know, a 60-to-1 carbon to nitrogen ratio. This rye on the left there, it might be at a 35 or a 40-to-1 carbon to nitrogen ratio. But if we just had, like, let's look at this cornfield. If we just had a high carbon load out in front of there, we're going to limit the amount of nitrogen available. So if we had to, like I said, we had to kind of understand this carbon to nitrogen for soil respiration as well. So this field here on the left is a rye field. It's about 4 to 6 inches tall. And that carbon to nitrogen ratio is about 14-to-1. So thinking about that ideal diet at 24-to-1, if we were to terminate this rye at this stage and plant corn into it, it's going to break down that grass really quickly. It's going to break down that rye. It's going to release those nutrients. Whereas if we plant into this 35-to-1, depending on our soil respiration, it's going to break down slowly. It's going to tie up nitrogen. So here's a cover crop of that or nutrient analysis of the biomass of that cover crop of that short cereal rye. And this is actually just taken last week. So we had about half a ton of dry matter. So not a lot. You're not going to build up a lot of soil with just a half a ton of dry matter out there and a 14.5 carbon to nitrogen ratio. But 36 pounds of nitrogen, about 36 pounds of potassium in there, and 11 pounds of phosphorus. If we were to look at this cereal rye that's more mature at about a 35-to-1, it's going to be slowly mineralized out and break down. We say, okay, how can I offset this? So, what can I do to make it so it doesn't tie up nitrogen, or don't have such a yield hit on my corn crop or milo crop? Well, we could apply nitrogen to that. And if we think about a campfire, and we back when we were younger, we thought, gasoline was fun to throw on fires, right? Well, if we throw a little bit of gasoline on a little bit of kindling on that fire, what's going to happen? We ended up, we got a great big boom out of it, and that's real fun, but we just primarily fed the microbes that nitrogen that they needed because they had all the carbon there. So we ended up with this charred piece of wood, but on the inside, it's still pretty intact. So the microbes just broke down what they could with that excess nitrogen. Now it's tied up inside of it and it's not being released, right? So what happens is we end up having, this rye residue that's still on the ground like we see here, but it's being tied up. It's not allowing that nitrogen to be released out. And so that corn plant or the milo plant, it's going to be suffering. It's going to need nitrogen. And it doesn't matter how much more nitrogen we apply on this, that residue is still a high carbon to nitrogen cover crop. And as soon as we apply nitrogen on there, we have to remember those microbes are the first feeders, and they have all the carbon they want. So they're really efficient at utilizing that nitrogen. They're going to go after it and use that nitrogen to help break down the residue, not necessarily feed feed that corn plant. Now, that residue is actually doing some really good things as far as helping to build that aggregation. It's going to keep the soil covered a lot longer than something that's a really low carbon to nitrogen ratio. So that's going to help to build the aggregation. It's going to build, this is a journey through soil health. So it's really going to build us the opportunity to earn the right to be able to start reducing our nutrients in the future. It's helping to suppress out some weeds, but it's probably going to inhibit our yields there. And that's where most people see, if they talk about planting corn into a rye field, they talk about, that just doesn't work. Another thing that I'd mention is, if we plant rye at 60 pounds an acre, let's say, versus rye at, let's say, 10 pounds an acre, well, what rate's going to take up more nutrients, 10 pounds an acre or 50 or 60 pounds an acre? Well, obviously the higher rates are going to take up more nutrients. So we have to understand that too when we're putting things into blends and ratios of what's going to tie it up and what's not. So you can't manage what you can't measure. And so when I start thinking about, how do we set the stage for what we do today to have a really good success this fall for planting our cover crops, if we don't have anything, any cover crops out there right now, or if we do have some, but we want better, we've got to take some steps here. And so the first thing is we really need to pay attention to our herbicide residual carryover. Certain herbicides like ALS herbicides, the PPO herbicides, some of the group 27s, HPPDs, and some others have some pretty long residual carryover. The HPPD is not really a thing. I guess there is balanced flex beans out there that you can spray a HPPD on. But generally speaking, after soybean harvest in most of the Midwest, follows that the next year with corn. So now we've got to look at like the ALS herbicides and the PPOs. Those herbicides are going to have the longest residual effect on a lot of our brassicas and a lot of our legumes. So we got to be careful when we're looking at what herbicide applications we're going to make here this spring and summer for what cover crops we're wanting. And the next thing is we need to plan for success. 1 day of growth in September is worth 5 days of growth in October. Just thinking about growing degree days, it gets cooler, the days get shorter in October. So if we're able to plant that cover crop in September, we're going to have more growth going into the winter. So we're going to have a better success the next spring with that cover crop. So we can earn the right to start reducing the amount of nutrients applied. So when we start thinking about that, we probably want to start thinking about planting soybeans first instead of corn. Corn's a warm season grass. Let that soil temperature warm up. Let's plant soybeans first and let's get them off earlier. The other thing is, let's plant a shorter day maturity or shorter variety of soybeans out there so that, you know, they go into reproduction a little bit earlier. We get them off sooner, get the cover crop in. I always stress, get the combine or the drill in the field with the combine. Chase that combine out with the drill if we can. The next thing we need to look at is our Haney test to determine our cover crop mixes. And I do this based on our soil respiration. You know, the lower the soil respiration, we probably need to be a little bit heavier on the legumes compared to grasses if we're going to corn. If it's going to soybeans, leave the legumes out. We can throw in Nebraska, but be heavier on the grass side. Now, if our CO2 respiration is high, now then we can start earning the right to throw in more of the grasses because we'll mineralize, break down that cover crop really quickly, and we'll be left with bare soil, which increases the amount of evaporation. Weeds might come through there, all kinds of compounding effects there. Allow that cover crop to mature as long as possible. So thinking about protein is 16%, or nitrogen is about 16% of a protein value. So thinking about alfalfa hay or something like that, if we were to have a very lush alfalfa hay out there and we cut it when it's short and only had, you know, 500, maybe 1,000 pounds of dry matter, but our protein level is really high, and let's say 30%, well, take 30% times 1,000, and I'll tell you how many pounds of protein that you have out there on a per acre basis. And then 16% of that would roughly be about how many pounds of nitrogen you would have. So if we allowed that to mature, yes, our carbon to nitrogen ratio is going to be maybe a little bit lower, or a little bit greater, I'm sorry, but the tonnage or the amount of total tons of protein, total pounds of nitrogen we'd have there would be greater as well. Once again, that all goes into context. You know, they're your northern environments and stuff. You can't allow things to mature too long. I like to utilize this cover crop biomass test, you know, at termination of planting and even throughout the year, taking either a 2x2 or a 3x3 square and taking all that residue off and sending it off to the lab to see what's in there. Couple that with a Haney test, a PLFA test. Repeat that at about every 30 days or roughly V4, V5 time frame when we're thinking about making a side dress application. Then we can determine how much nitrogen do we actually need to come back with. Take it again at tassel time. Then we can figure out, you know, how much is going into that plant. And then again, take it, if we have that biomass still there, I would recommend going out at the end of the season, about black layer time on corn, and take another biomass sample and another Haney sample. Then we can start to understand, if we did this every year, we'd be able to trend line basically where our nitrogen is, where our soil microbial activity is, how much is in the leaching out of that cover crop and going into the soil, and how much of it is being taken up by the plant. And are we becoming more efficient or less efficient with our cover crops and our use of nitrogen there? So here's an example of this 3x3 square taken in the cover crop. So we need to look at, you know, we've got this low carbon and nitrogen cover crop on the left, which is going to be a quick release of nutrients, versus this one that's slightly higher, which is a little bit slower release of nutrients. So it's going to be, the one on the left is going to mineralize out fast, and the one on the right is going to more or less immobilize a little bit longer. It will mineralize out, but it's just going to take longer. So on these two fields, interesting enough, the CO2 respiration is both 16. So considering, you know, thinking about microbial activity in the soil, 16 is pretty low. So the one on the left, it had that cover crop had a 10 carbon to nitrogen ratio, while the one on the right had a 19. So thinking about what's that mean? So if you think about the microbes in our soil, if we've got this low carbon to nitrogen or this low CO2 respiration, think of that as a very immature system yet. Think of it like a baby, right? We wouldn't feed a baby a steak and potatoes. That looks really good to me right now, but you know, the baby's going to be really efficient with milk and some smashed up food, some baby food. So that low CO2 respiration is going to need that low C to N cover crop for that high C to N cash crop. Now, as we get into a more matured system where we have, you know, maybe these little babies turn into athletes, and now they're on a team of you know, a bunch of their buddies playing football, and they come over, why they can eat you out of house and home. So basically now we've got something that's kicking. We've got something that's a higher CO2 respiration rate, and they can consume more of that higher carbon to nitrogen ratio cover crop without basically tying it up. They can help to release it and cycle it. So again, there's our carbon to nitrogen ratios for CO2 respiration. So the first one, these are backwards. So the first one, where it's number 1, is actually that 19.2. So it's the higher carbon to nitrogen ratio. It would have been that image that was on the right. The second one it had a 10.3 carbon to nitrogen ratio. So we took these samples at the end of May, and 30 days later, we checked it again. And what we've seen is the carbon to nitrogen ratio at 19.2 with that 16 CO2, it only released out about 52 pounds of nitrogen, whereas with that 10.3, we released out 147 pounds. Well, the thing about corn, and we'll see here in a slide, is that corn utilizes its nitrogen much later in the growing season. So we had a rapid release of nutrients out front. Could have been a bad thing, you know, but you know, results all vary here. So those are 2 things I want to focus on. Another thing I want to focus on is this potassium. Notice how much of this potassium was released in the first 30 days. Potassium in that plant is 100% water soluble. So as long as you catch rain, it's gonna come right out of that plant tissue. And the amount of phosphorus that was released out of here, and calcium too. So this second biomass sample had a lot more vetch in it. So what I noticed out of that is that phosphorus and the calcium in that vetch was able to be mineralized out and then cycled out of that residue relatively quickly. Whereas where we didn't have the vetch, the hairy vetch, it didn't cycle out as much. So what that equated to, if we were to take that 147 pounds from the second sample, if we were to look at what it, you know, on a $20 cover crop mix, $30, whatever, it's basically almost $274 worth of fertilizer value right there. So again, in Southern Illinois, we have the advantage of letting things grow. So we've talked about the principles. You know, we're keeping that soil covered. That's not a roller crimper. It's just a brilliant roller. Just knocked it down. The nice thing about vetch is anything vetch climbs up on, it drags everything down when you hit it with anything. So you can see the rye in here as well. And so we rolled it down, planted it, and there's that corn coming right up through the vetch. Yeah, you can see there's some skips out here. Some guys might harp on a few skips and, oh, that plant was a little bit later. You know what? Who cares at the end of the day? You know, so here's a difference in the first Haney test to the second. You know, our CO2 respiration, it increased. Our water extractable organic carbon increased. Percent MAC increased. So we see here the Haney test nitrogen. We started out with 15 pounds of nitrogen in the soil, and a month later, basically, we had 53 pounds of nitrogen. So at V5 time, we took some tissue samples on corn. We had never applied anything. This cornfield right here never had anything applied to it. It was a small field and we thought, what the heck, let's do what we can do. So at V5, everything came back looking really good. At VT, at tassel time, still everything was looking good. A little bit low on magnesium, but everything, and boron, but everything else was good. So here's an image of that field, satellite imagery. You can see it's surrounded by a lot of timber. The deer absolutely love this field. They clean out everything. The only thing you have is anything in the inside. One of the cool things that we seen when we started looking and analyzing our corn plants is our brace roots had this mucilage on it right here. So basically that corn was releasing these root exudates out of these brace roots, which has a microbe in there that's able to take the atmospheric nitrogen and convert it and make it to become plant available. So now companies, seed corn companies, are trying to figure out ways that they can make corn plants produce their own nitrogen. What we find is when we limit the amount of nitrogen that that plant has, we see mucilage. Here's that plant, again, about tassel time, not seeing any type of deficiencies out there. And here's what it was at harvest time, 170 bushel corn. Another cool advantage, so this farmer, we'd switched to 38-inch rows. And so since we delayed termination until everything was well senesced and a hard seed, as soon as that canopy opened up, he didn't even have seed cover crop. Everything just came up. The oats, the rye, vetch, clovers. He had a solid standard cover crop that he didn't even need to seed out in that field. So let's talk about economics here a little bit. So on this field, if we were to look at the traditional acre in that area, conventional, if we were to figure on $200 cash rent and 3 tillage passes, plus all the fertilizer, the herbicide, and the harvest, plus hauling, plus crop insurance to get 170 bushels, and this was in 2020, we'd have been losing $33 an acre. Well, on the regenerative side, without any fertilizer being applied, but we did have to apply cover crop. We did roll down a cover crop, so we had that expense in there, but everything else remaining about the same, you know, we made $214 an acre. So yield being the same, we made a lot of money. But I often hear guys say, well, you know, you plant corn into cover crops, you're going to give up 20 bushels yield. Well, even if I give up 20 bushels like I'm showing on this slide, I still made $132 an acre. So I could either lose $33 or I can make $132. I don't know what your banker would say, but I'm pretty sure he'd be happy if you made $132 an acre. So then I also hear guys talking about baling off corn stalks and selling that. So we had to understand, okay, what's in that residue that we're removing off that field. So it's roughly about, you know, on 170 bushel corn is about 2.8 tons of residue. So that's about 4,500 pounds of carbon, 45 pounds of nitrogen, 16 pounds of phosphorus, 113 pounds of potassium. So that ends up being, you know, a nice $219 an acre that we're removing in nutrients right there. Not only that, but we're also removing all that habitat for some very beneficial insects that can become predators in our soil systems as well. I hear a lot of guys talk about slugs. Well, if we don't create the habitat for the insects that consume slugs, we're going to have slugs. It's pretty simple. Okay, my computer froze up just a second here. All right, here we go again. So, well, go back. We need to look at also how do we manage in-season nitrogen with our corn or milo or anything. So this is an example of a corn crop and looking at when nitrogen is being taken up and where does it go? You know, between the leaves, the leaf sheaths, the stalk, the tassel, the husk, the cob, and then into the grain. So first thing I want to notice is here at about V6 time frame, which is about 30 to 35 days after planting, depending on growing degree days, units. But that's usually when that corn plant starts to utilize nitrogen. You know, prior to that, it's taking up phosphorus. After about this time frame, that plant doesn't take up a whole lot of phosphorus. So we have to say, you know, where are we applying our nitrogen? Too many times we're out here, especially in the I states, you know, we're applying nitrogen all the way back in the fall for this cash crop, 6 months in advance. Where's that nitrogen going to be? Even with N-Surf, can we guarantee that the nitrogen is going to be there for that crop? Or how much are we just exporting down to the Gulf of Mexico? The other critical point that we need to realize is here about R1, so about VT to between R1 and R2, so basically when that kernel gets a blister on it, that's when nitrogen uptake ceases. You know, now it's taking nitrogen from other pools inside the plant to fill up into the grain. So we have to understand when to apply, how much to apply to be able to get maximum yield out of this for that corn plant's potential. But if we're putting it all up front, we're subject to big loss. We put a little bit up front, it's about 10 pounds of nitrogen per leaf stage on corn. So if we were to do it with the planter, you know, we could maybe put 60 to maybe 100 pounds on. We can come back in and we could side dress here and take those right measurements, figure out how much we need to apply to get up to here. So in season nitrogen management, you know, we've got 100% fall applied or pre-plant versus a split application. This is a big no. We don't want to see this. You know, if possible, let's put a little bit out front, but let's put the majority of it out in the crop when that crop needs it. There's different tools looking at, and this is just a few examples. You know, this culture knife, this works really good in a clean tilled field like this, maybe a straight no-till field, but with residue, you'll get it balled up right here within the first 50 feet of going in the field so bad that you will be mad at the world. This isn't much fun. I don't recommend this kind if you have heavy residue. This kind of system right here that just dribbles it on right behind that disc opener works good. This is a Y drop system here that dribbles nitrogen right at the base of those stalks. Works decent as long as you're in a humid environment or an area that you can guarantee that you get the moisture whenever you put nitrogen on that soil surface. The nice thing about the Y drops too is if it's on a sprayer, you have the opportunity to come in basically all the way up till tassel time and apply nitrogen. So you could put on 5 pounds a week if you really wanted to, and really spoon feed that crop. Just takes a lot of time and a lot of energy. So another thing I notice is oftentimes is a lot of talk about, you know, is there anything I should put in with my nitrogen? Well, if I think about this anhydrous applicator, and anhydrous is about 82% nitrogen, right? So that's a high concentration of nitrogen we're putting into this system that's actually a lower carbon or lower nitrogen system. So what we end up doing is oxidizing off or burning off a lot of carbon out of that soil profile with anhydrous. But the thing that I want to focus on is this 10-to-1 ratio. So our soils are about this 10-to-1 ratio. And ironically, if we were to look at our like corn, wheat, milo, it's roughly about a 10-to-1 ratio or a 12-to-1 to 15-to-1 ratio of nitrogen to sulfur as well. So generally I just say, hey, you know what, feed it the same percent that the soil sees it at. I don't know if that's right or not, but in my mind, if I was a microbe, that seems a little bit, you know, more stable than just straight nitrogen out there that could be tied up. You know, we're not, the nice thing is with all the clean energy acts and everything and clean air, we don't have as much acid rainfall. So, you know, we've taken a lot of sulfur out of the environment, but at the same time, we need that sulfur from that acid rainfall for our crops, which we did see in that soil profile. We do have a lot of sulfur in our soils. We just usually aren't able to cycle it, usually due to the lack of carbon and due to the lack of microbial activity in that soil profile. But, you know, if we're to do anything, putting some sulfur with the nitrogen definitely helps to utilize that nitrogen. The sulfur in that plant is basically helps that plant to tell through the messenger RNA what to do and how to assimilate those proteins that Shane was talking about in the nitrogen as well. So that was kind of a quick overview and run through. to let y'all know, here's some of our upcoming webinars we have. Next one will be with our continued biblical webinar with Dr. Alan Williams and Sam Bass. Also Regen Ag 101, Soil Health Academy, adaptive grazing courses, and we'll start Q&A.
All right. Thank you, David, Shane. Excellent. So I'm going to dive right into the questions here. The first question, Shane, was for you on your presentation. Chad's wondering, the biomass sample you showed, what was the carbon to nitrogen ratio of that?
I'd have to go back to look. I think it was a 19-to-1, if I remember what the seed-in ratio was on that. That was annual, or that was cereal rye. And I didn't acknowledge, but there was no nitrogen put down in that system. That was planted in after corn. So it was just, we took a clipping to find out what the nutrient tie-up was, and that cereal rye was. I was blown away. You know, no nitrogen being provided to this system. You know, I was just kind of emphasizing, you know, how much of that organic fraction we can actually convert back into the inorganic form to be utilized by the plants. The big thing is, and David talked about this, you know, we look at the Haney soils test, and we could probably do a whole series just talking about nutrients and talking about, you know, on that Haney soils test, looking at CO2 respiration. You know, basically, the more the microbes we have working, especially the more diversity we have working, the more of these nutrients we're going to be able to cycle back into the plants and into the vegetation.
Thank you, Shane. Ike asks, how fast will and what plant stage will the legumes be when they can put out nitrogen so that other plants will benefit from them? David?
Yeah, so when a lot of us, when we think about legumes, we think about where the nitrogen is being produced. So we start thinking about in that root, right? Because that's where that rhizobia bacteria is going to fix that nitrogen through the through the Krebs cycle of taking carbon and phosphorus to be able to split that dinitrogen bond. But I challenge everybody, that's a very, very small percentage of nitrogen when you look at the total amount of nitrogen in that plant. It goes back to talking about even the grass being a carbon to nitrogen ratio. As soon as we have any biomass above ground, there's nitrogen there. So any of that biomass in that legume growth, there's nitrogen accumulating in there, because think of it as an animal feed, as protein. And there's protein there, there's nitrogen there.
Very, very good. Donald asks, if you start regenerative farming with very diverse cover crops, say 20 species on the first few years, can you go down to just a few species, say less than 5, for subsequent years, rotating through different species each year? Shane?
Well, it's going to depend on your environment. You know, it's going to depend on the time of year you're planting, how many growing days you have. You know, obviously we know things function well with diversity. You know, and when we do diverse mixes, you know, we kind of look at it a little bit of a lot. So we can take advantage, you know, sometimes it's environmental things we may be dealing with. Some cover crop species are more drought tolerant, dry, versus others. Definitely, I'm thinking more of cover crops after a small cereal like wheat in my part of the world. You know, I like the diversity. I like to stay, like I said, with like I said, I want to be feeding different soil microbiology, different root architectures, you know, getting down to that soil profile. I mean, like with those tests, we look at the total nutrient test. You know, we're only looking at 12-inch soil profile. Think of all the nutrients are down 24 inches or maybe even deeper. You know, I want to keep utilizing the biology along with the plant, cycling those nutrients out of my system, and, you know, getting that cycle functioning, that nutrient cycle.
I think it is important here, though, David, perhaps you can talk about the importance, though, if we're going to roll a cover crop, we have to take into account at what stage those covers are going to be in during rolling, so we may not be able to have the diversity. Would you explain that a bit?
Yeah, so when we're looking at rolling cover crops, and if that's our termination goal, we need to look at plant species that basically are maturing about at the same time. You know, for instance, if you had winter barley and cereal rye in the mix, that winter barley is going to mature way earlier than what, you know, the cereal rye would, which becomes a little bit of a challenge. And even with some of the legumes, there's several different legumes, whether it's vetches or clovers, that have different maturities that may roll and crimp well, and others that will just pop right back up. So always try to focus on ones that complement each other.
Great. Here's a question. If you have been rotating cover crops, cash crops in a regenerative way for years, can you eventually get a volunteer cover crop without planting it on some years? Or can you reduce the seed per acre in a significant way to your cover crop so that you will still provide the armor for the soil? Shane?
Yeah, well, I've tried this. I've tried to allow some of my cool season cover crops to go to seed and planting into them and thinking that seed will germinate and I'll have a cover crop without I have to do another drill pass. I have yet to have much success with it. I've seen some legume seeds, warm season stuff like hay beans that people have let go to seed and they come back up voluntarily the next spring. You know, what I'm concerned with is you start building up the soil microbiology, you know, you're going to start going through residues really quick. And I'm probably more concerned that you're going to have a lack of armor if you don't have that plant population in order to keep that soil armored up.
Perhaps though, David, you want to explain to them what we saw at our friend Daniel's though out in California just a couple of weeks ago.
Yeah, absolutely. And I see this often too, is that certain species will come back, you know. Some's going to depend a little bit on your context of your rainfall environment, you know. Some of your cereal grains and things like that, if you're in a high rainfall environment, they may rot, you know, whereas your hard seeds like your legumes, especially the vetches and that, they'll come right back. You know, we're seeing in California, yes, Daniel, he does do some irrigation, but, you know, he had some oats and stuff like that that volunteered back. And just like in that picture I showed that, you know, some species do. Now, as we start to, especially keeping our soils moist and keeping it covered, you'll start to have maybe more crickets and things like that. And those little guys can be ferocious eaters of seeds. So I wouldn't get in the habit of thinking that it's going to be there every year, and to just plan on seeding something if you're going to do annuals. Otherwise, switch it to a perennial basis.
That's a good point, David. And you mentioned granivores. Granivores are insects that eat seed. Dr. Randy Anderson, ARS Brookings, South Dakota, did some really good research on the amount and percentage of seed that will be consumed by these granivores in a healthy ecosystem. So I would say as your system advances and you have a healthier system with a balance of biology, those granivores are going to consume a lot of that seed. Our good friend Willie has a question here and a statement, and he raises an interesting point. And then Shane, I'll ask you to expand on it a little bit. Willie wants to raise the point of tracking the nitrogen from the source, which originates from the atmosphere. It can only become part of the biomass and ultimately become part of the soil organic nitrogen when incorporated into plant biomass, if fixed from the atmosphere, and not only from legumes, as there are thousands of free living nitrogen fixers depending on the root exudates from grasses. It is also not only NH4 and NO3 that is a nitrogen plant food source. Water extractable organic nitrogen presents a larger nitrogen food source in a stable ecosystem. The point is that the nitrogen component from the biomass has to be accounted for which is from the atmosphere. Shane?
Yeah, it's a correct statement. I mean, well, it does bring up some good points. I guess, there are a lot of free living nitrogen fixating bacteria. The unfortunate part, a lot of that is a lot of systems are lacking that. That's the reason when we talk principles, we're trying to start building the habitat back to build back that soil microbiological community. That biology is, moving by wildlife, it's moving by air, it may be also within the soil system, just waiting for the conditions to be right in order to become active again. Unfortunately, as I think we can all acknowledge, a lot of our systems, and I'll even acknowledge my own system, we started working with degraded systems. This is a journey, as we've mentioned, about moving down. But the only way we're going to get down this path is following the principles. There's no ifs, ands, or buts about it. I mean, if you're truly wanting to start getting a nutrient cycle functioning and utilizing what you have, there's only one way, and that's it.
Yeah, you're exactly right. And Willie raises a very good point about the water extractable organic nitrogen in a stable system. That's going to be the main source of nitrogen. We'll see it converted from inorganic to organic. And I think of it as a slow release system over time. That's why we see perennial pastures that, although they test extremely low in inorganic nitrogen, they test extremely high in organic nitrogen. Good points, Willie. Thank you. Kenneth asks a very good question here, David. On new land, would you go cold turkey and not fertilize and then plant cover crops? Or would you put synthetic fertilizer on as a starter?
So I guess it'd be, what are you comfortable with? If you're comfortable looking at, depending on your cash crop or whatever you're trying to grow there, there's goals for soil for grazing, I guess. I was just down in Mississippi looking at some ground here a week ago, 2 weeks ago, and they cut that, it'd been corn cotton for years, and they quit and went straight to a cover crop. And they were really disappointed in the growth of the cover crop for the last couple of years. I use the analogy as kind of like an alcoholic or somebody addicted to drugs, that you take that addiction away from them and they collapse. If you cut it out cold turkey, get ready for some bad days. And that's basically what our soil's going to do. So our soils, are a living ecosystem just like we are ourselves. And that synthetic fertilizer is basically that drug. So, you can cut it out cold turkey, but, just know that you might not see the greatest growth, or it might look a little bit ugly. And if you're okay with that, then that's fine. The biggest thing is, that I see in a lot of it is we don't allow things to mature enough, especially in the situation like we're trying to graze here. It's easy, I guess, there, but keeping that soil covered with that mulch, not just that living when you're looking down and seeing the green vegetative, but keeping it covered with that mulch layer from the previous year's residue and everything is extremely crucial of building that aggregate. If we think about how do we build an aggregate by keeping those principles in place, then we start to earn the right to cycle more of these nutrients. Now you can start by keeping the first year or two, having a very low carbon and nitrogen, depending on your CO2 respiration, but having maybe a lower carbon and nitrogen cover crop mix. And then the next cover crop that you put in there be a higher one. So you can cycle that back through.
Yeah. As a rule, Kenneth, if you've been using synthetics a long time and that have not been applying the principles to that field, you'll probably want to add some small amount of fertility so you can grow some biomass and jumpstart that biology the first year or two. Next question is from Michael, and he asks, Do you think biological products that fix nitrogen for corn are a good source of nitrogen to wean away from synthetic forms of nitrogen? Who wants to take that one? David?
I'll start. There's all kinds of these products out there on the market today that are claiming that they can give you 40 pounds of nitrogen. I've yet to see the product trials where they actually reduced the nitrogen by 40 pounds when they added the product in to show if they actually got a yield increase or not. The other thing is, most of these products, you don't know if that microbe in that jug is alive or not, you know? And it might not even be for what your soils need. And if you've been going down the path of soil health and you add these microbes in there, you're probably not going to see any benefit. Now, if you're a conventional tillage guy and you add it, maybe you'll see some benefit. But we also have to look at what's the main carrier other than water in that jug. And the majority of it's humic acid. And so basically that's being a stabilizer for that. You know, if you're applying it with especially some of these products that you mix in with starter fertilizer, that humic acid is basically stabilizing that nitrogen to both forms of the cations and the anions, and it's going to slowly release that nutrient out. So are you getting it? Are you getting the benefit from the slow release of nitrogen, from the nitrogen you're applying, or from the bug with the humic? I'd have to question that.
Thank you, David. Grant Bright Kreitz asks, is there anything other than synthetic stabilizers to hold synthetic nitrogen stable for any length of time for release in corn? David, do you want to take that?
Yep. So just like I mentioned earlier, humic acid would be one of them to look at. Basically, it binds that nitrogen. It's not a big misconception if somebody's out there telling you it's a food source for microbes. It's not. It's already been degraded down to the point where fungi can't even utilize the carbon in it anymore. So being that humic acid is derived from lithodite coal beds, it's very highly degraded down already. So basically that carbon is a binding site for the cation, the anions, your nitrates, and the ammonium in that to be able to slowly release the nitrogen throughout the growing season. I think it is University of Kentucky has some research with humic acid that actually showed that you could reduce nitrogen by 30% with humic acid and not give up any yield.
Great. An attendee asks, any chance of having a webinar on reading and understanding a regen soil test? I believe if you go to the Understanding Ag website, go up under resources, look at past webinars, you will see we have done webinars with Lance Gunderson on just that topic. So I suggest you start there. Caleb asks, I understand that long-term monoculture legumes, alfalfa, etc., really hammer soil aggregation. My question is, how high can we go with legumes in a short-term cover crop mix ahead of grain sorghum or wheat without a lot of deleterious effects? They're asking 70-30 legumes, grasses, 80-20, 60-40. Shane, start with you.
It's going to depend a lot, Caleb, on your CO2 respiration, on how much legume component we really should need to put into that mix. So that's where I would start looking at that. You know, it's also going to depend on when your termination is, like David talked about, the carbon and nitrogen ratio when you terminate that cover crop ahead of grain sorghum. So there's going to be a few things I want to look at. You know, obviously, you know, grain sorghum's a little different than corn when it's really going to need its nitrogen, the nutrients. So we're going to look at a little bit different carbon to nitrogen ratio, termination ahead of that. I know David's done some work with grain sorghum and cover crop termination.
David, anything to add to that?
Yeah, a couple things. So, and I noticed Luke in the chat had asked a question too about alfalfa or monoculture legumes and the detriment to the soil. And one of the things that we see if it's long-term monoculture alfalfa is just that alfalfa actually doesn't make the soil any more mellow. It actually makes it extremely hard. And we'd seen that, a couple of years ago down there in South Central Kansas when we were trying to pull some pans to do a rainfall simulator on three-year-old stand alfalfa, that we had to use a sawzall to cut the soil to get it in the pan. It was so hard. So if we think about alfalfa, like I said, these legumes require a lot of nitrogen, but to produce nitrogen, it requires a lot of carbon, a lot of phosphorus to break that nitrogen bond. So they're not putting back any carbon back into that soil system. So if, I just wanted to address that. The other thing is, just like Shannon mentioned, it's all going to depend on your CO2 respiration, but results may vary. We got to read some fine print here too, that it depends on your rainfall, what your rainfall environment is, of what you can and can't afford to do. In a higher rainfall environment, you can have a higher grass component because it's going to mineralize out a lot quicker than in a lower rainfall environment as well. So, to tell you, to be at a 70, 30, 80, 20, 60, 40, 50, 50, it's all going to depend on what we see from, that Haney test and what our goals are. Some of the other things that we could do is delay termination, in a mix if we have the grasses in there. Especially, I know where you're located at there, Caleb. So, if it looks like you're getting really dry or whatever, or if you have too much grass in that mix, possibly, that you could early terminate some of the grasses and let the legumes continue to grow. Could be an option as well.
Okay, thank you, David. Shane, what cover crop mix do you use after wheat harvest in your area?
I like sorghum sedans. I like all my grass species, millets. We'll do a combination of different legumes, maybe a little bit of sun hemp, hay beans. We'll put in maybe just a dash of a sunflower, buckwheat, maybe add a pound, if the most, of a radish into those mixes. It's going to depend mainly on my resource concerns. So if I'm needing more armor, I'm probably going to go a heavier grass mix. If I'm needing to cycle a little bit more of that armor, that cover on top of the soil, I might spike up a little heavier on the legumes. And, also how am I going to capture an economic gain? Am I going to stockpile this? Am I going to use it through grass finishers? So there's a lot of things that, that allude to the mix we put together.
Adam, I would also add to that, don't forget, depends on your herbicide use in that wheat crop, because you want to make sure nothing carried over there. David Chase asks, I've been building cover crop mixes online. It seems the diverse mixes become cheaper than the lower species mix. Does this normally happen?
So I guess by lower species mix, he's just saying straight up legumes, maybe, is what I'm guessing. Yeah, so the legumes are usually expensive. When you look at like the hairy vetches and the clovers and stuff like that, they're probably the most expensive part on a seeds per pound that you would put in a mix. But at the same time, there's more seeds per pound, so it doesn't require as much. So when we were talking like in that question previous, a 70, 30, 80, 20, whatever, 60, 40 blend, we're looking at that on seed count, not by a pound basis. So yeah, when we start to mix things together, we have to remember that we're planting things and we want them to succeed. So you're not going to do 50 pounds of cereal rye and 20 pounds of hairy vetch. That's going to break the bank, but you can back these rates back and it's going to be a lot cheaper. You got to think about how many seeds in that per row foot are you placing and how many will successfully grow. Too many times, especially in warm season mixes, we see a lot of guys, they'll go really heavy on some of the grasses and they've got legumes in there too, some cowpeas or mung beans, and they never see those. Well, the grasses get off growing so quick that they shade it all out and you don't ever see it. So that's the advantage of being balanced at that. And yes, it will be a cheaper rate. Long story short.
On that note, David, Donald asked, as regenerative agriculture catches on, is there a risk of cover crop seed going up in price with increased demand?
Absolutely. So not only that, but we also have to look at what's going on in the environments where a lot of this seed is being produced at, right? So think about cereal rye, barley, oats, triticale in the last couple of years, really, but really last year was bad. But the drought in Canada, North Dakota, Montana, really hammered those areas. And then you look at like where a lot of the vetches and the legumes, clovers are being produced, and ryegrass as well, is in Oregon. Well, last year they were hit with a drought, and the year prior to that, during harvest, after they swathed a lot of the clover and vetches and stuff like that, they kept getting rain, so their seed quality was poor. So we went into 2 years in a row now with a limited seed supply, add trucking cost into that and fuel surcharges. We have got to start figuring out how do you raise cover crops locally. If you could take out just a few acres of your farm to plant cover crops on, you will be very well money ahead. I can promise you that.
And not only that, David, I would add to that with rhizophagy and the ability to proliferate biology on that cover crop seed. If it was grown in your local area, that's a benefit as well. Zach has a question here, and David, I think you covered it, but I will make sure. Zach says, I recently read that adding humates in with nitrogen application helps keep soil health more functional and helps with the plant uptake and allows a guy to apply less nitrogen. Is this accurate? What in the world are humates? Thank you.
Yeah. So, humate can just be the breaking down of that residue. Those carbonic acids start to produce and make humic acids right there in humates. But simply what you see from whether it's a box brand Monties or whoever out there selling some type of a humate, there's a lot of companies, like I said, they're trying to sell it as some type of a biological soil health product, a wonder world, one size fits all. Apply this, it'll cure all your problems, and it's good feed for your biology. That's a lie. Microbes can't break it down any further than it already is. But what it does for nitrogen is, yes, it helps to bind it up.
Okay, Martin asked, any experience with Johnson Sioux compost extract to replace chemical fertilizer in fields that have been managed conventionally or recently converted to regen? Shane?
Well, no, I don't have any experience. I have done the Johnson Sioux, I guess, we've inoculated seed with it. I would do some testing prior. I would really encourage doing the Haney soils test. Let's see what's in there that we have within the soil system first. I'm not saying the Johnson Sioux is maybe not practical, but I'm not going to sit here and say that's going to replace 100% either of chemical fertilizers. When I'm looking at stuff, everybody wants to go down the regenerative path, and I applaud anybody and everyone to do that. What we've got to understand is, we've got to grow biomass. In order to provide armor, we've got to have biomass. So that may mean at the early initial beginnings, I may have to still utilize a synthetic form of nitrogen in order to help grow that biomass. But then I'm going to be very intentional from that point forward, making sure I have principles in place and trying to build that system back. But I'm going to test. The best thing I can do is maybe try a small plot of the Johnson Sioux compost extract and see what it does compared to the rest of the field.
I would just say that Dr. David Johnson always makes it a point to tell everyone that Johnson Sioux is not a replacement for synthetic fertilizers. It is a way to proliferate biology. Martin, I would encourage you to go to the Understanding Ag website, the resource tab, and look at the past webinar that was given by Brandon Bach. Brandon Bach uses Johnson Sioux compost extract on his operation, and he's been very successful with that. He is still using a much reduced rate of synthetics. Brandon Bach webinar. Mark asks, is there anything more I can add to 28% plus sulfur to help break down my wheat stubble and dead cover crop residue to help get those nutrients to the corn crop faster this spring? David?
I would say, why do you want to do that? Because then you're going to have bare soil so quick that now you're going to have weeds that pop up. And if we're adding nitrogen and sulfur to break down residue quickly, I mean, that's just an added expense. I can guarantee that by the time you plant into that, within a few weeks, it's probably going to be gone already if it's starting to decay and process. As soon as the planter rolls through it, if it's not laying on the ground, it will be on the ground. And as soon as it has direct, as soon as that plant residue is right where those microbes are, the soil, this is why rolling and crimping will decompose residue quicker than residue that's staying upright, is it's down there with those microbes. They're going to come up and they're going to start eating on that and decomposing it really quick. And with the rainfall, especially Mark in your area, you're not going to have a lot of residue. I think I'd hold on to my 28 and use it for in crop.
Okay, Greg asks, would you recommend planting a cover crop mix into an old alfalfa field the fall prior to planting a cash crop like corn? Shane, there's some unknowns there you'd have to have answered, correct?
Yes. There's several unknowns, but it's something, I would probably consider doing. It's going to depend on your area. Obviously, like David talked about carbon to nitrogen ratios. I'm gonna go back, and also talk about the Haney. I'm gonna pull that Haney soils test. I also want to pull a phospholipid fatty acid test. See what's kind of going on underneath within the soil itself. It's going to depend on my mix. I mean, obviously, an old alfalfa field, a lot of nitrogen, high. We're going to have probably pretty collapsed soil structure. I'm going to probably come back in with a very high carbon, crop like cereal rye, wheat, something like that, triticale, into that alfalfa mix. And then, depending on what my stand looks like, kind of my carbon to nitrogen ratio, when my termination will be on it.
Yep, a lot of questions. Donald asked a question and I will answer it. Is cover crop seed grown on farms conventionally or regeneratively? Donald, no doubt the majority is grown on farms conventionally. However, we encourage you to seek out those that grow it in a regenerative manner. And the goal is that more and more will do just that. Mr. Breitkreitz asked, is there a difference between technical and food grade fertilizer compared to conventional fertilizer sold at most co-ops? David?
So most of the food grade fertilizer is sold as a low salt fertilizer. Well, if we were to look at 28 and 32% nitrogen, we would look at that and say, well, 28% is actually technically a low salt fertilizer. It's all salt. It just depends on how much is in it, to say one's better or worse, I mean, that horse has been beat so many times. It's tough to go into all a Q&A session here, Grant. But is there a difference? Some, yes. Especially if you're using it as a pop-up starter fertilizer. Some of the, if you were to use like 28% as being a low salt fertilizer and you put that in furrow, you're going to kill germination. Some of the 1034Os out there, if it's a nice Kelly green color, good. But if it's brown, and coming from maybe, I don't know, an old foundry or something like that they used for engine blocks or something, that's not going to be real good stuff. It's going to be pretty hard on seed germination. So it all depends on what you're trying to achieve with it.
Thank you, David. Question from an attendee: as we move to a biological system with corn, should we look at eliminating putting 28% N on with the planter and only side dress? David?
I think that would just depend on one, your environment, what cover crop are you growing? How big can the biomass get? But yeah, I mean, if you're putting it on with the planter, the nice thing is, like I said, roughly that corn plant is going to use about 10 pounds of nitrogen per leaf stage. So, I would consider split application anyway. So put a little bit with the planter if you have the system already, and then put the rest on with side dress. If you don't have that on your planter, just wait and do it as a side dress application. I do know that some guys try to do a weed and feed with cover crops out there after planting, and they try to spray a 28 application with their herbicide. Basically, those microbes are taking that 28 that you spray, and they're using that to help break down carbon. You're not really feeding your corn at all with that. So, it's one of those things that you're going to degrade your residue really quick. You're probably not getting any benefit out of it too. We're looking at return on investment to bushels.
Okay, here's our final question for the evening, and I'll take a swing first and then turn it over to Shane. Mitchell asks, is the end goal to completely red all applications of nitrogen fertilizer, or is some degree of input going to always be necessary? And Mitchell, I'd say that totally depends on your context. I, for one, firmly believe that by and large, we're trying to outproduce our environment. Most environments, you can only cycle so much carbon. And this day and age, we're being told yield, yield, yield. And in order to get increased yield, pushing the synthetics. And I think there is some long-term negative ramifications for doing that. On my own ranch, we have not used 1 pound of either synthetic or organic nutrients besides those that drop out of the back end of an animal since 2007. And our yields are consistently higher than county average. They're not near as high as some in the county, but those come at a cost. So you have to look at your context and what you want, and then design your program accordingly. Shane?
I think you addressed it really well, Gabe. And the comment you made, sometimes we're trying to outproduce our environments, I think is a very accurate statement. I mean, it's going to kind of hinge back to that. It's also going to depend on what you're comfortable with. Do I fully believe we can get back to probably a fully functional system? Yeah, but it's going to take time. We got to remember working with an ecology. We're going to work at the pace of that ecology. It's also going to depend on, what your production goals are. There may be times you're still going to require some synthetic nutrients being applied in order to achieve those. You may be trying to, outproduce the environment to a degree. So, I mean, I think there's a lot of, like Gabe said, context we put into that of what you want to do and where you want to be. It is, like Gabe, we haven't utilized any synthetic nutrients on any of our crop ground. We'll make that stipulation, or my pasture land. We still have to utilize synthetic nutrients on our hay ground. That's because we remove all that biomass. We're not cycling that biomass back through the system again to capture those nutrients. So it's all up to your comfort level and, like Gabe said, your context.
Thank you, Shane. Thank you, Shane and David. Excellent presentation this evening. On behalf of Understanding Ag Soil Health Academy, we want to thank everyone for joining us here today. Be sure and check out our websites. We have some upcoming academies and BDA Farm in Alabama, at CNS Ranch in New Mexico, number of good academies coming up throughout the year. And then also take a look on the Understanding Ag website for upcoming webinars. Due to the fact that it's spring, we get most everyone's in the field getting seeding, planting done. We're going to back off on the webinars a little bit, but we will be picking them up again once we get through spring's rush. So be safe out there and take care, everyone. Thank you.